Today's lecture, we're going to have a discussion about the human host and disease transmission. And these principles that we're going to discuss are very essential in relationship to disease prevention and disease treatment. So, in terms of human host and disease transmission, we're going to discuss the basis of human disease, and we're going to examine disease in relationship to the disease triangle. We're going to talk about non-random distributions of disease. Why, for instance, are only some individuals infected and others are not? We're going to talk about the different ways that diseases are transmitted between individuals. We're going to talk about how the symptoms of disease can help us determine disease causation, but we're also going to see that some diseases don't even have any symptoms. And that poses a very special challenge that we'll discuss in detail. We're going to talk about occurrence of disease, in which populations and in which vehicles, and what are some of the risk factors for these diseases. We're going to talk about measures of disease transmission. We're also going to present an example of a disease outbreak that occurred about 5 or 6 years ago in 2008, the Salmonella in the tomatoes outbreak, which many of you may remember. So, what is the basis of human disease? Well, human disease is really a function of the interaction between the host, an agent or agents, and the environment. And these vectors, I have an example here of a mosquito. The vectors are organisms, usually insects, that can transmit the agent to the host through the environment. So basically, the host needs to be susceptible. That, for instance, if the host is immune to the disease agent, or if the host has antibodies, or if the host even has a vaccine against the disease agent, then the host will not be infected. But barring that, host may be susceptible if, for instance, they've never been exposed to that agent. Now, the agents must have, very important, the capacity to infect. So some agents don't have that capacity, or some agents, if they're eradicated or if they're weakened, for instance, then they can't infect the host either. And then the environment, of course, functions to both promote infection or prevent infection, and also the environment can serve to change host susceptibility. And some examples of that are inclement weather, or, for instance, malnutrition or unavailability of water, or, for instance, too much water, or a disaster even. Then certainly that alteration in the environment can certainly promote infection of that disease. So what are some of the reasons that diseases only occur in some individuals? Number one, genetic predisposition. A great example is breast cancer, that women who carry the BRCA1 gene, or a mutation in the BRCA1 gene, are at very high risk for developing breast cancer. But there are genes that increase or decrease susceptibility to other infectious diseases. A great example is malaria and sickle cell anemia, that carriers of sickle cell anemia are basically protected from malaria. And we'll discuss that example when we get into parasites. Nutrition. If you are malnourished, your susceptibility to infectious diseases is greatly increased. On the other hand, if you're well fed, then your susceptibility is less. You can be immune compromised. If you're immune compromised, then of course your likelihood of being infected is greater. Your socioeconomic status. Poor individuals in general worldwide are at greater risk for infectious diseases or for developing infectious diseases than wealthier individuals, and this occurs worldwide. Disease, now, there's a seasonality element to diseases, and they change over time, but they also change over years, over decades. So we can see a rise in incidence of an infectious disease. For instance, we develop great antibiotic agents. If it's a bacteria, the disease incidence lowers, the infections go down, and then we can see reemergence. So our first lecture in epidemiology presented several examples of diseases that have reemerged. So for these reasons, and there are certainly others, but these are the main reasons. For these main reasons, disease and health is not randomly distributed. So what are some of the ways in which diseases, infectious diseases, can be transmitted? Number one, direct transmission. So airborne, we can catch diseases by inhaling water droplets from other individuals who carry influenza, for instance. Blood, certainly we can develop bloodborne. We can become infected by sharing blood products, sharing needles, through sexual contact, all sorts of ways in which we can actually have become infected from diseases based on those factors. Also water. So for instance, if you're swimming in a pool and someone else has an infection, a skin infection, for instance, you can have that bacteria transmitted to you through the water. There are a number of ways of, a number of modes of indirect transmission, one of which is a common vehicle where individuals share, for instance, a door handle or a towel or even food. So we're going to have an example of a foodborne infection where basically food that was contaminated is eaten by individuals, and then certainly they can become infected that way. You can have a single one-time exposure, or you can have multiple exposures, or you can have continuous exposures, and we'll talk about that in more detail later in the lecture. Now, the other important indirect transmission vehicle are vectors. So vectors are generally insects, and they transmit diseases between individuals, and they can also transmit diseases between humans and other animals. So certainly, and we're going to see several examples of diseases which are transmitted by mosquitoes, for instance, but we also have the tsetse fly, and we have several other kinds of parasites and things in which in which the disease agent is basically reliant on the vector in terms of transmission. And there was one time way early in the AIDS epidemic that it was thought that the AIDS epidemic was transmitted, or the AIDS virus was transmitted by mosquitoes as well. So what are some examples of disease transmission? So a great example are the different behaviors, right, that increase the risk of becoming infected by the AIDS virus. So for instance, in all adolescents and adults, and then we have the data according to males, and then the data according to females. And you can see that distribution of methods by which the disease is transmitted are very different for males and females. So, for instance, males, 67% of transmission of the AIDS virus is due to male-to-male sexual contact. Now, in women, the primary way is through high-risk heterosexual contact. So, for instance, if men are bisexual, right, then they can certainly spread the AIDS virus to women. And also high-risk heterosexual contact as well, and women who engage in sexual practices with men who are at high risk, who also have high-risk sexual behaviors, can catch the AIDS virus from men. Injection drug use is actually less common in men than in women, And certainly sharing needles is a primary way in which the AIDS virus can be transmitted. So these are basically behaviors that individuals engage in, but they're also ways in which the disease can be transmitted. So for hepatitis A, B, and C, we can see there are differences according to which hepatitis variant or which hepatitis type it is. So hepatitis A can be spread by food. So you can, but it can also be spread by sexual contact. So we see that injection drug use is pretty uncommon for hepatitis A. Homosexual activity among men is more common. International travel is a very significant way in which you can contract the hepatitis A virus, and suspected food or waterborne outbreak as well. Now, I recently traveled to India for research, and I had to receive a hepatitis A vaccine because of the international travel, and also traveling to an environment where hepatitis A is more common than in the United States. Now, hepatitis B and hepatitis C are blood-borne viruses. So you can see injection drug use is the primary way in which hepatitis C is transmitted, and multiple sex partners. Now, the baby boom generation are thought to actually have a potentially high prevalence of hepatitis C virus that perhaps is dormant because of the practices that occurred in the 60s and 70s, and we don't have a vaccine for hepatitis C. So they could, so the certainly is a screening of the baby boom generation is being seriously considered by the government, and it could be that routine testing of hepatitis C among individuals, gosh, about age 50 or older, may become more commonplace. And also, we also have a vaccine for hepatitis B as well. So hepatitis C right now is a concern among practitioners, certainly in relationship to potential susceptible populations where we don't even know that indeed they're infected because they don't show symptoms. And we're going to see later how important that really is. So let's talk about clinical and subclinical diseases. So certainly if a disease results and is characterized by signs and symptoms, and those symptoms and signs are characteristic of that disease, then we know pretty much with a certainty that that person has been exposed to that pathogen. Now, but we have other instances of subclinical disease where, for instance, if you're in the early stages of disease, you don't yet show symptoms. So preclinical disease is a term that's applied to diseases that the individual is infected, but at that point in time, they don't yet show clinical apparent disease or signs or symptoms. And this is very important because those individuals are potentially infectious. They can spread the disease, and they don't yet know indeed that they have been exposed to that disease. So, and in those individuals, so that term is really applied only to the individuals who are destined to progress to clinical disease. So these are the individuals that have been exposed, and they will go on to eventually develop the disease. And we know that, for instance, with HIV, right? That you can be exposed to somebody who has the AIDS virus and get yourself tested. But what's recommended is that you wait for 6 months because that's potentially how long it takes for symptoms to appear. But also sometimes that's how long it takes for the antibodies to appear as well. So that's really important in terms of disease detection. Now, there's another category called subclinical disease, and these are diseases that are not clinical apparent and they're not ever going to be clinical apparent. And these situations are often diagnosed by serologic antibodies, okay? So an antibody response. So say you've been exposed, but you didn't show any symptoms. Maybe the degree to which or the level or the titer or the amount of virus or bacteria you were exposed to was very, very low. You never showed any signs, but you do have antibodies. And by having antibodies, that will indicate that indeed you have been exposed to that particular pathogen. Now, we also have non-clinical disease. Well, then you're wondering, what is non-clinical disease? Well, that's having a disease without any signs or symptoms, and there's several examples of that. So one example is persistent disease. So this is a disease where we have infections lasting for years and sometimes over the lifetime. And a really good example of that is the chickenpox virus, and then the shingles that later appears, 20, 30 years later as shingles. And this is another potentially emergent disease. Right now we have vaccines for smallpox, and now we actually have vaccines for shingles. But this virus, basically what happens after the original disease course of chickenpox is finished, right? The child gets over it. That virus actually migrates into the cranial nerves usually and lives there for years until the individual, through age or through stress or whatever, if they become immune suppressed, for instance, that virus can reemerge, and then we have the shingles symptoms appear, which certainly can be catastrophic. So we also have examples of what's called latent disease, and these are infections with no multiplication of the agent. And basically in that case, usually what happens is the viral DNA fully integrates into the host genome, and basically there's no more evidence of the virus, but the viral genome has been fully integrated into the host. Now, the final example we're going to discuss is the carrier status. Now, the carrier status is a very important category that often, for many reasons, goes undetected. And these are individuals that harbor the infectious agent, but they are considered not infected. They don't have any evidence of the antibody, and they don't have clinical symptoms. However, these individuals can be infectious. So, and this status, right, can be limited, or it can last for a long time. So a very famous example, Typhoid Mary. Typhoid Mary was a carrier of Salmonella typhi. She caused many typhoid outbreaks, but she did not have clinical symptoms, and she passed away in 1938. She was a cook, and she was a live-in cook. She cooked for households in New York City, and she moved from household to household. So we all think that she probably knew she had the disease, right? She's trying to hide it because, she kept changing her name, right? And, she wanted to, obviously have a job. So Typhoid Mary caused 10 outbreaks with 51 cases and 3 deaths. Now, there are in some instances, these individuals can actually be, if you can imagine, incarcerated, right? Because they are that infectious, and we've had examples of individuals who willfully infected others with the AIDS virus, for instance. I remember one case where they actually imprisoned the guy because he willfully infected an individual, and it was determined that that was the case. So, you know, that's possible, right? But this carrier status is an important classification, especially in relationship to being infectious and yet not showing signs and symptoms, and also not even showing any antibodies. So what about the different ways in which disease occurs? What are the patterns of disease? So one pattern is called endemic, and that's defined as the habitual presence of a disease within a given geographic area. Now, this concept is very important because what in one area might be an endemic disease, in another area would be considered an epidemic. So a primary example is, for instance, malaria. Excuse me. So malaria is endemic to the tropical countries in Africa, also in Asia. And so, for instance, if you travel to those countries, it's recommended that you actually take prophylactic agents or medication that can actually, excuse me, can actually manage the disease in the event that you get it. Excuse me. Now, epidemic, of course, is the occurrence of illnesses that are in excess of normal expectancy. So I'm going to show you a very common example that we're all aware of. Pandemic is a worldwide epidemic that across the board is considered an epidemic. And examples every year are the influenza. So right now we're all concerned about H5N1. And on a yearly basis, we tend, in certain times of the year, of course, everybody goes out and gets a flu shot because at that time, right, the whole world basically is experiencing the influenza disease occurrence. And so that's a great example of a pandemic. So the obesity epidemic, at least in the United States and other parts of the world, are certainly is not really an infectious disease, but it's considered an epidemic. And you can see here in the year 2000 that the prevalence of obesity was much lower, was basically 20% or below in the United States. But the last, 10, 15 years, the numbers of individuals who are obese has greatly risen. And in fact, there's a number of states now, you can see here in 2009, these darkened states have more than a third of the population are obese. And recent figures, I believe, show that Mississippi now has the highest prevalence of obesity in the United States. So, and Colorado, boy, Colorado, they're great. They keep much lower than the rest of the United States. But basically, and you can see just everywhere, there's more and more people becoming overweight and obese. And, you know, certainly policy-wise, we need to make some serious changes in the ways in which we are all living our lives. Now, what are some measures of how disease is transmitted? So these measures are important to understand because they actually give you clues in terms of what disease is, for instance, causing these symptoms. So a lot of times, especially in the beginning, right, you don't know what disease individuals have. And so you have to basically trace back and try and figure out how did they get the disease, what caused them to get the disease. But one of the first ways that you can actually figure that out is through the incubation period. So the incubation period is really the period between infection, right, by the disease agent. So the time in which the disease agent enters the body, and then the appearance of symptoms. Okay, so that's certainly important in relationship to what the disease actually is, because these incubation periods are very characteristic. So basically, there are many determinants of the incubation period. So there's some variation in relationship to even incubation periods of the same disease agent. One determinant is replication. So how quickly does the infectious agent replicate, right? How quickly can they colonize? How quickly can they reproduce? How quickly can they create more organisms? Because the more organisms you have, right, the more likely you're going to have symptoms. What is the site of where in the body did the infectious agent enter? Where in the body does the infectious agent replicate? So an example, the measles virus, right, enters the body through the respiratory system. It's an airborne virus, and the virus eventually resides and replicates in the skin. So, you know, but there are other examples too, where, for instance, if the site of entry is a long way away from the actual place or the part of the body in which the disease agent resides, then, you know, the incubation period will be longer. Another example is tetanus. Okay, the tetanus bacteria. Remember, say you step on a nail with your foot, right? And the tetanus agent releases toxins that are neurologic, right? So the distance between your foot and the brain is probably the longest distance in the body. So that might be that incubation period for that individual might be longer than, for instance, if you cut yourself with a rusty nail on your hand, for instance, and that incubation period would be quicker. And then finally, what is the dose? You know, if you take on a lot of virus or a lot of bacteria, if you're infected with a high dose, right, like the anthrax spores, my God, if you got a high dose of those, right, then your incubation period is going to be shorter as well. So that length of time is very important, and again, it's characteristic of the particular agents. Some agents take a long time, others not very long at all. So the next measurement, right, of disease transmission is the epidemic curve. And that's really a very simple curve where you just figure out at what time periods individuals become, where, symptoms appear, right? And then you can also use that to trace back and figure out the incubation period, because with the incubation period, if you can figure out when the pathogen or when the individual first got exposed to the pathogen, then at that point in time you can see what it is that they did. For instance, what kind of foods did they eat at that time? That could then give you clues in terms of what the disease agent is. So what are some characteristics of a disease outbreak? So one example is being exposed one time to one agent with a common vehicle. So, and this is a situation where all the cases develop by exposure to the same agent. So, for instance, a particular pathogen carried in food. So I would say on a weekly basis, we hear in the news all these different foods that are contaminated that get pulled off the shelves. And these particular lot numbers, cans of food, canned during certain times, at certain time periods, they have certain ID numbers, and they all get pulled off the shelf because those foods carry that particular agent. So in this situation, the exposure occurs only once. The outbreak is generally explosive, so generally a lot of people get the disease. And again, remember in disease transmission, what are the reasons why some of these things are emergent? Transportation. We have a wonderful transportation system in relationship to food distribution. So we've got food on the shelves all across the United States. You know, everybody eats particular item that's contaminated, and all of a sudden you have thousands of people that potentially are consuming contaminated food. So in those situations, you have a sudden and rapid increase in the number of cases, and the cases are limited to people who share that common exposure. So another way that they can determine this is by looking for very characteristic strains. So when a particular food is contaminated with a particular strain of bacteria, then when you go to determine, for instance, who has been exposed to the food, all you need to do is, for instance, take a blood sample and measure whether or not these individuals have antibodies to that particular strain. And then because a lot of people develop food poisoning, for instance, but you want to know which ones got food poisoning because they ate a particular food. So the other criteria about this concept is that secondary transmission is rare. So what does that mean? That means individuals, for instance, who primarily become exposed by eating food, they don't then expose other individuals, right? They themselves just remain primarily as the individuals who got exposed. Now, on the other hand, the exposure can be one-time exposure, the exposure can be multiple, or the exposure can be continuous. Now, we can see examples of a one-time exposure, but a multiple exposure can happen because they don't yet know that this particular food is contaminated. And so, for instance, if the source remains undetected, people keep consuming the food over and over again, especially if it's a commonly consumed food. So say for instance, we suspect an outbreak. What are some analyses or what are some questions that we need to ask? So one question is, who is being exposed? Who is getting the disease? Now, I mean, that seems pretty obvious, but again, like in food poisoning, for instance, there's a lot of people who get food poisoning, but who are the individuals who are getting poisoned because of a particular food that's contaminated? And if indeed we can isolate and figure out who those individuals are, then we can trace back and figure out what they ate. And also, given that we know now what they ate, we can, look at that food, we can study the food, we can extract whatever bacteria might be living in the food, and then we can, conclusively demonstrate that's the case, and then basically put out a recall and take that food out of the food supply. Place, that's another one. So we have person, place, where was the case infected? Did the case happen to go to a particular restaurant? Did the case, did it occur in one particular town, in one, area of the United States? Knowing where the case occurred or where the case was infected will also help us determine what the cause of the disease is. And then finally, time. When did the infection occur? And remember, we can often evaluate that in relationship to measuring the incubation period. So in terms of these foodborne illnesses, the CDC estimates 76 million cases of foodborne illness per year, 325,000 hospitalizations, and 5,000 deaths. Now, this data was as of 2008. I would suspect it is much higher today. But the other thing that we have to look at is also our ability to detect these diseases has greatly improved. But also, a lot of foodborne disease doesn't get reported. Oh, you get sick for a day or two and then you just carry on, right? And so a lot of people don't even know, for instance, what food caused their symptoms. And some people, if they have healthy immune systems, they don't really suffer as much as infants and also the elderly. So we only know the pathogens for only a fraction of the individuals that are hospitalized or that actually, seek medical care. And, 62 million, so most of these cases of foodborne illnesses are from unknown agents. We don't know what caused them. Okay. So let's talk about an example of a foodborne outbreak that occurred in 2008. And perhaps some of you remember this. It was quite a huge undertaking by the CDC in 2008. And it took quite a long time to conclusively demonstrate A, what was the food that caused the contamination, and B, what was the agent that caused the disease. So I'm going to, take us through this example, but it's not the only food outbreak example we're going to see. We're going to see a couple other ones in our later epidemiology lectures. So, in June 25th, 2008, and the reason why actually I have this data is because I was teaching a class and I happened to go online to the CDC at that time. So at the time I went online, the disease was very early. And they were starting to observe a particular outbreak strain of Salmonella that they thought, at the time when I picked up this data, they thought at the time that Salmonella was being actually carried in tomatoes. And after quite a bit of study, so I'll take you through this investigation, you can see that it actually wasn't just tomatoes, but at the time they thought it was tomatoes. So here you can see that Texas and New Mexico were the states that had the highest number of cases. And, California had some, Arizona and Illinois were also other states that had quite a few cases. So later in the year, later that summer, I went back in and I thought, I'll look and see what's going on with the disease now. And so then I picked up this new data, and you can see that there were many more cases that had developed. So New Mexico had more, Texas had quite a bit more, and now Illinois had quite a few as well. So, as the disease was progressing, the investigators were closing in on tomatoes, and at some point, they actually recalled tomatoes. So at that time, if you remember, they completely removed tomatoes off the shelves in the markets. They took tomatoes out of the restaurants. They were completely out of the food supply for, quite a bit of time. So this is our epidemic curve. And so in May, the CDC got notified. The first multi-state studies started actually, within a week. Then in June, we had a national tomato alert. And then we had a second multi-state study. And then finally, in July, we had a jalapeno pepper alert. And then finally, so you can see, so let me explain. So when tomatoes got removed from the market here, time subsequent, or time after that point, we still saw cases. So this told the investigators that removing tomatoes off the market did not completely eradicate the disease. So we still observed cases, although they did drop certainly in frequency, but we still observed cases until finally jalapenos were taken off the market, and then the disease went way, way down. So that's one hallmark. So we'll talk in later lectures what some of these hallmarks are. So if, for instance, you remove the food out of the food supply and the disease drops, then you know you found the right agent. And sometimes you don't fully know the agent even, but as long as you know the food that is causing the disease, if you remove the food out of the food supply and you eradicate the disease, that is a great, that's strong evidence that you have basically suppressed or cured, so to speak, the particular food outbreak that's going on. But in this case, we still needed another, 2-3 months before we finally fully determined what the foods were that were carrying this particular bacteria. So the other thing that you want to ask during the time is if you have candidate agents for what you think might be causing the infection, then you actually characterize the symptoms of that particular agent. So for instance, Salmonella causes diarrhea, causes fever, causes abdominal cramps as soon as 12 to 72 hours after the infection. You can identify the pathogen and the infection by stool samples. The illness lasts 4 to 7 days. Now, for healthy individuals, sure, it's an inconvenience, but among susceptible populations, it can be deadly. So, for instance, the elderly, immune-compromised individuals, and infants, we had a number of individuals die from this infection because they were very susceptible. Now, the severe cases of Salmonella happen when the Salmonella bacteria spreads from the intestines to the blood supply and then can actually infect other sites. And we actually have a discussion about this in our infectious patterns lecture and also in our bacterial morphology lecture and several other places. We do discuss what happens or how do these bacterial infections how do they become more serious? So these are various studies that were conducted to identify which foods were carrying the bacteria. So, and we're going to learn actually how to conduct this particular type of analysis in our other lectures. So the thing to look for here are odds ratios, and we'll learn in another lecture what an odds ratio is. But an odds ratio that is very high, so an odds ratio of 62 is extremely high. So as an example, the odds ratio of lung cancer for a cigarette smoker who smoked, 30, 40 years is about 20. And here, this odds ratio is 62. This is a really very strong association. So this was found among a Texas restaurant that served salsa. Okay, and that's a much larger odds ratio, for instance, than some of these smaller ones. But so an odds ratio, if an odds ratio is greater than 1.0, that means that's a positive association. And so you can see even in these other places a moderately large odds ratio. But we see here that in Minnesota, right, we have an odds ratio of 62 for jalapeno peppers that were available in this Minnesota restaurant. We have a reasonably high odds ratio for guacamole, right, among, in a restaurant in North Carolina. So, there was another study done of two different kinds of peppers, and they were actually both pretty similar. But the reason for the complexity of these results is really because tomatoes are often eaten together with jalapeno peppers, so it became very difficult to separate out the two foods. So, but we saw in the epidemic curve that removing tomatoes lowered the incidence a little bit, but not that much. So it was only after removing the jalapeno peppers that the epidemic was basically over. So in terms of describing the investigation, tomatoes, and what the investigators think, was that tomatoes possibly were a vehicle for infection in the beginning of the epidemic or in the beginning of the outbreak. And later studies identified an association between eating raw jalapeno peppers and Salmonella. And we know, for instance, that tomatoes and jalapeno peppers are often eaten together in salsa, right, and in guacamole, both. So, but when they actually localized the investigation to jalapeno peppers, they did what's called a traceback investigation. So we're also going to discuss that in great detail in a subsequent lecture. That particular finding was sufficiently specific enough and strong enough that launched a traceback investigation where they employed these various techniques to trace back the jalapeno peppers to Mexico, which ended up being the source of the outbreak, or the outbreak bacterial strain. So, our first question is, given what we learned in lecture about disease, what are some ways we can thwart transmission of the disease? Well, I'll tell you. Number one, we can alter, both the agent, the host, and the environment. And if the disease has a vector, we can alter, right, the vector. Okay, and what I mean by that is by altering, we just alter disease transmission through these various factors. So, for instance, with the agent, right, what can we do with the agent? We can give a vaccine, or we can give pesticide, or we can give some kind of treatment. But really, this vaccination is really what we can do to the host, for instance, to protect them from the agent. Pesticide, so, if, for instance, the agent is, living in a, so, we can also use a disinfecting agent, disinfectant. And then in treatment, so for instance, if we're able to treat the host, right, and cure the host of disease, then that particular agent is no longer, considered to be a disease agent or considered to be an agent that we are concerned about. So in terms of the host, I talked about certainly developing vaccines for the disease, improving immunity, right? Improving host immunity. Through better nutrition, okay? We know, for instance, if you have a malnourished population, they're very susceptible to infectious disease. So some of the research I'm doing is actually learning how to use nutrition as a way to strengthen the immune system's response to these various diseases. The other thing we can do is actually alter the environment, right? So we can do things like provide netting, right, for mosquitoes. we can actually, a better case here is we can use pesticides to eradicate the vector, okay? You know, we can certainly also improve sanitation, right? That will certainly serve to protect the population from being exposed to cholera, for instance, if we improve sanitation. So these are just some ways that we can thwart transmission of the disease. And sometimes, you know, we actually have to do an environmental change, right? Like you know, unfortunately, you know, pesticides is really a rather poisonous approach. But in relationship to malaria, for instance, if you can find a pond, if you know of a pond that's where mosquitoes live that are infected with Plasmodium, for instance, using pesticides will certainly eradicate them. But also providing the village with nets, for instance, will also help greatly to prevent their exposure to these mosquitoes. So our second question, why do you think it took so long to identify the Salmonella Saintpaul outbreak? Number one, tomatoes and jalapeno peppers, they are eaten together. And because of that, it was very challenging to distinguish, you know, which it was. Was it tomatoes? Was it jalapeno peppers? But one of the real keys to that outbreak was when they took tomatoes out of the food supply, we still saw cases, which suggested that it was another food. But in that case, you know, sometimes you have to go through these steps in order to figure it out. You know, another reason may be that there were a lot of subclinical cases where the individual's symptoms were not all that serious and they weren't reported probably in the beginning. So, you know, and sometimes, and then the challenge, if you can imagine, of identifying the source of the food. So, for instance, doing that traceback investigation to find that the jalapeno peppers were in Mexico, right? You have to evaluate all these different suppliers, all these different transport mechanisms, and all these different places in which grow jalapeno peppers, for instance, and being able to trace back to the final location of where that salmonella came from was very, very challenging. And again, we're going to see a couple other examples in some of the other lectures, and you're going to see the challenges that current food scientists are facing, or food safety investigators are facing in relationship to identifying all these pathogens. It's very complicated. So the third example are, what are some of the ways you can detect subclinical cases? Number one, antibody detection. Now, once the test for HIV once the test for antibodies to the AIDS virus was discovered, okay, or basically was discovered, at that point, then we could go back and see which individuals had antibodies to the AIDS virus. And a lot of individuals didn't even know, for instance, that they were carrying the AIDS virus, and the only way they knew was actually by getting a positive AIDS test. So especially, I was telling you about that window of infection. So with the AIDS virus, right? You need at least 6 months, right, for antibodies to appear. So if you get an AIDS test somewhere in here, if it's negative, it doesn't necessarily mean that you're HIV negative. So what they recommend is you get the first test and then you wait for 6 months and then you get the second one. And if the second one's negative, then you know, I mean, you have a much greater chance of being negative. So the number one way really is antibody detection. But you know, in the case of Typhoid Mary, she didn't have antibodies to typhoid, right? She didn't have any antibodies. And so in that case, what they did was they just kept seeing that every house she ended up going to, the individuals came down with this infection, right, with typhoid fever, and then they were able to finally conclude that indeed Typhoid Mary was the cause. And in that case, they deduced that information by evidence. But another way, if possible, you can directly identify the pathogen, but that's very expensive and it's very difficult. It can be very difficult. A lot of times you really need to know what you're looking for. You can't. It's very challenging to just, you know, go blind and try and find something, right? Usually you need panels and various things to test your serum or blood against these known panels. And, you know, certainly it's better to start when you at least have your antibodies. So that concludes our lecture about the human host and disease transmission. Thank you so very much for visiting educator.com.